Hi guys, Miss Francis here. Today we're going to talk about control of the cell cycle. So by the end of this video, you should know these vocabulary words: checkpoint, cyclin, CDK, MPF. If not, please rewatch the video, go over your notes, come for extra help, read your textbook. So the cell cycle control system. How is the cell cycle controlled? We don't want our cells to continue indefinitely dividing, so we need some sort of control system. So embedded in the cell cycle are 3 checkpoints, where these checkpoints are critical points where a stop signal or a go signal helps regulate the cycle. The 3 major checkpoints are G1, G2, and M. Sometimes it's called S. So let's revisit what happens at each one of the phases during the cell cycle to see what might happen at each checkpoint. In other words, what might we look for at each checkpoint in order to successfully move to the next checkpoint? Well, at gap 1, that's when we're experiencing cell growth. So some things we might look for in the G1 checkpoint are there nutrients? Did the cell increase in size? The S phase or synthesis phase involves DNA replication. So at the G2 checkpoint, we want to make sure that our DNA did in fact replicate. Then in the G2 phase, you're going to have your finishing of your cell growth. Your spindles are going to form, and you're really going to prep for mitosis. So at this at the M checkpoint, you're going to make sure that you have spindles, and you're going to make sure that your chromosomes are in fact attached to those spindles, so that when cytokinesis occurs, when the cytoplasm separates, those spindles are holding onto those chromosomes, pulling them to either side of our new daughter cells. So here's just a review if we went over. The G1 checkpoint checks for cell size, for nutrients, for growth factors, and for DNA damage. The G1 checkpoint is, in most cells, the most important checkpoint, because it's at this checkpoint that certain tumor suppressor proteins can play a role. In other words, if I have DNA damage, I don't want that cell to continue dividing. So a tumor suppressor protein known as P53 can initiate a delay in the cell cycle in order for that DNA to be repaired. Furthermore, let's say I don't want that cell to continue dividing. I can tell that cell to enter a resting state, or G0, where that cell no longer divides. Now, some cells can be called out of the resting state. For example, liver cells can be called out of the resting state and return into the cell cycle. However, cells like nerve cells, cardiac cells, those cells will not be called out of the resting state. Those cells don't divide. So here's what I just said, that the non-dividing state is known as the G0 phase, and certain cells can be called back to the cell cycle. Those cells are called back by growth factors, but there are other cells, highly specialized cells, that don't ever get called back, so to speak. They never divide. So how exactly is this cell cycle regulated? It's regulated by different protein signals, which either inhibit or activate the cell cycle. Molecules called cyclins, together with CDKs, or cyclin-dependent kinases, kinase, remember, enzyme, form MPFs, or maturation-promoting factors, where the proteins are going to regulate the cell cycle and cause proliferation. So how exactly do these molecules work? Well, the concentration of the CDK, the cyclin-dependent kinases, remains constant throughout the cycle, but the concentration of the cyclins increases and decreases throughout the cycle. Once the threshold of the cyclin-CDK complexes are reached, then that cell cycle is told that it can start the next phase, either the S phase, or it can progress through the S phase, or it can start the M phase. So how can these cell cycle regulatory proteins be used in cancer treatments? Well, inhibitors of CDKs can be used to treat cancer. So RB is one of those tumor suppressor proteins. RB is retinoblastoma. When an unphosphorylated retinoblastoma binds to E2F. E2F is a transcription factor that's going to turn on genes in the nucleus. So when retinoblastoma binds to E2F, that's going to stop cell growth. It's going to give the stop signal at G1. Furthermore, P16 acts as a tumor suppressor protein because it binds to the cyclin, thus preventing the cyclin CDK complex from forming. Therefore, it stops at the G1 checkpoint because that threshold of CDK cyclin complexes is never reached. The other tumor suppressor protein that's highly studied by scientists is this P53. So what P53 is, it halts the cell cycle at G1 checkpoint so that the DNA repair system has time to fix that damaged DNA. And the way it does is it initiates the transcription of P21. P21 binds to the cyclins so that the cyclins cannot bind to the CDK, thus inhibiting the cell cycle at the G1 checkpoint. Now, how do we even know that the cell cycle is controlled through signals? Two different experiments were done. The first experiment involved fusing a cell that was in the S phase with fusing a cell that was in the GAP1 phase. And what ended up happening is that cell in the GAP1 phase immediately entered the S phase and synthesized DNA. In the second experiment, a mitotic cell, or a cell that was in the M phase, was fused with a cell that was in the GAP1 phase, and the GAP1 phase cell immediately entered mitosis without duplicating its DNA. So it seemed to skip over the S phase. And from that information, what scientists took was that there must be some molecules present in the cytoplasm that control the progression to S and M phases.